Our next speaker is Amy Foreman, and Amy is a plant ecologist for the Environmental Surveillance and Education Research Program at the Idaho National Laboratory site in eastern Idaho. She leads a research and monitoring effort addressing sagebrush steppe land management issues, including long-term vegetation dynamics, plant community structure and classification, post-fire vegetation recovery, revegetation with native plant species, and patterns of non-native spread. She has a BS in biology from Idaho State University, where she studied the emphasis of plant community physiology and quantitative ecology.
All right, thanks, Bryce. And Tom, I want to thank you for having me here to talk about this particular research. Throughout the course of my career so far, I think this is probably my favorite project, and I've had the opportunity to work on some really cool projects. So hopefully over the next half an hour I can get at least a few of you as excited about population biology of sagebrush as I am. So it's just a bit of a background. About 10 years ago we started a conservation management planning effort at the INL site. And this is basically their strategic effort to put together a monitoring plan and a way to identify natural resources and put together a comprehensive approach to managing those resources. So as part of that effort, we sat down and figured out what our conservation targets may be. And of course, we had several native plant communities and native plant species. And at the INL site, much of the site is dominated by big sagebrush, and Wyoming big sagebrush in particular. So that's one of our more important conservation targets. And when we started looking at what was going on with those species at the site, we saw that we had two different issues. Of course, big sagebrush is declining there, as it is across much of the West. And we could divide those declines into both acute losses and chronic losses. So these are our acute losses. This is a fire footprint map. It looks at fires from 1994 through current. And we've lost about 38, almost 40% of the INL site. Plant communities have been changed due to fire in those last about 20 years. And these acute losses are pretty easy to wrap your head around. One of those fires, the Jefferson Fire, burned over 80,000 acres in the span of an afternoon. So those acute losses are pretty dramatic, and they make an impression. And ultimately, we know the cause of those losses. Now, the dynamics that lead up to that, there's certainly still a lot of debate about, but we know we lost sagebrush in that footprint, and we know it was from fire. Chronic losses, on the other hand, are something that we hadn't thought a lot about, and it turns out that they're pretty dramatic as well. This middle line here is big sagebrush, again, mostly what we think is Wyomingensis. When we started collecting long-term vegetation data in the 1950s, average cover across our long-term vegetation plots was about 15%. In 1965, it hit a high of around 20%. And in 2006, it was only about 7 or 8%. Now, these are all plots that are unburned. So from 1950 to 2006, we see a decline of half. If we look at the difference between its high in 65 to 2006, we're at a third of what our cover was. And this is about the 60% of the site that hasn't burned. So we're actually seeing more sagebrush loss by these gradual declines than what we had seen from those acute losses. And these have happened over the span of multiple ecologists' careers, so they're not something that are as dramatic, and they're certainly not as easy to wrap your arms around. And more importantly, we don't know the cause. So as part of our conservation management planning effort, one of the things we're tasked to do is to put together strategies for managing our targets, for maintaining the health of our targets, for monitoring in a way that we can figure out what's going on with our targets, which way they're trending, and why. So it became more important at that point, it became more important at that point to figure out what the potential mechanisms were driving these chronic losses. And some of those listed up top. There's lots of information in the literature. Herbivory from both insects, all the way up through ungulates are possibilities. There are times when we have very fast snow melts and lots of spring rain creates anoxic soil conditions. We have die-off from those sorts of conditions. We have diseases, rusts and molds and things like that. And we also know at the site and across much of the West we had a large sagebrush die-off in the 70s. And so some of the low numbers we're seeing now may still be legacy effects from those die-offs. But as we work our way across the site, which it's about 900 square miles, we can see places where there are effects of herbivory. We can see low-lying basins where maybe anoxic soil conditions were a problem. Occasionally we can see where we may have had some disease impacts, but these weren't widespread. They certainly didn't explain what was going on across such a large area. So we moved on to these next set. Move on to this next set here of potential causes. Maybe we're seeing a reduction in fecundity, or a general loss of vigor, or maybe just a general decline in stand condition. And these are all more population related factors than maybe some of those first ones listed. And as we're looking at strategies for managing big sagebrush, it kind of struck us that we didn't know a whole lot about the population biology of the species. We think it's maybe a slow-growing, long-lived species. We often think about it as a climax community, a reference state, an end state, if you will. Recently, a lot of big sagebrush researchers have been talking about sagebrush as a foundation species, which is, I think, is an excellent way to sort of think about its role in the ecosystem. And it can even be considered an autogenic engineer in that once it's established in a stand, it changes the microenvironment around it for itself and for other species. And I talked through some of these with some friends who were foresters, and I asked them, when you want to manage a stand, when you have an issue with the stand not being in a condition that you want, what do you do? And it came to this idea of, maybe big sagebrush is analogous to some tree species. And foresters have certainly done a lot more work in population biology. And when they go to manipulate a stand, one of the first things they want to know is what is the age status, the population status of that stand. So that seemed to be a reasonable question for big sagebrush as well. So if maybe we could characterize stand structure of big sagebrush, and maybe even if we could find a tree species that had a similar population structure, we could come up with some management strategies that may be effective. So we put together a population biology study. The first objective of it was to simply sample big sagebrush stands and age them and characterize those stand distributions. Next, we wanted to figure out how stand age relates to the condition of the stand. Is there anything about its age that we can see? If a stand looks decadent and old and wolfish, if you will, does that tell us something about its age? And the third one here is to kind of explore the dynamics of stand replacement in the absence of fire. This research, again, was started almost 10 years ago, and at that time we had a lot of management recommendations from outside agencies asking that we consider some controlled burns. And we've tried that at the INL site, actually with very little success. It turns out that either it's going to burn a lot or it's not going to burn at all, so it's something we approach with caution. But thinking about it in terms of stand structure, burning or mechanical reduction or things like that, that may be analogous to something you would do in lodgepole pine if you had something you were wanting to maintain like that. Or is big sagebrush more of an old growth type of system where gap dynamics is more important in population turnover? So we picked 14 plots, each 1 hectare in size. We spaced them for the most part at least 1 kilometer apart to try and avoid autocorrelation. And we specifically selected plots to try and grab a range of condition, of age, of cover, of density, of abundance of other shrub species. And we used a stratified random sampling design. We ended up sampling 50 live shrubs and 20 shrubs that were dead. We wanted to know how old they were when they died. In each stand, we also sampled sagebrush density. We thought we were being clever, so we sampled this variable we called sagebrush condition rank. What we wanted was a rapid assessment technique to go out to a stand and be able to figure out a way to rapidly characterize its condition and maybe its age structure. So we put a rank scale on this. So a rank of 1, you would rank a shrub 1 if it were alive, or no, I'm sorry, 1 if it were dead, 5 if it were completely alive, 3 if half the canopy was dead. And we randomly sampled 100 shrubs in each plot and then tried to correlate it with the other variables there to see if it was a reliable indicator. And then we also did line interception for all shrub species on the plot. And these are our plot locations. The scale's kind of irrelevant. The two farthest plots away from each other are about 12 kilometers, and the two closest actually ended up being about 500 meters apart. And we chose those two that close because we were really trying to fill that continuum of condition, if you will. The vegetation map background here was actually completed about 3 or 4 years after we had completed sampling for this particular study. And we had 2 of our plots show up in things that we had mapped with that we were fairly certain were Wyoming big sagebrush stands. And this is really cool that I get to follow Bryce, because this big area where most of our plots are is a spot where we threw up our hands. For those of you that have been out at the INL site, we have large areas like this where you have low-lying areas and you're pretty sure you've got tridentata, and then you've got some areas where Wyomingensis seems to be what's there, and then there are a whole lot of stands that are really intermediate, and that's what we ended up sampling most of, were those intermediate stands. So after data were collected, we ended up with 915 slabs. The way we had collected slabs on the site, we followed basically the methodology put forth in the 60s by the Tree Ring Lab. We'd go out, wrap a wire around the base of the shrub, cut it off right at soil level, take about a 10-meter slab, take it into the lab, sand it 3 times with 3 different grits. We would use vegetable oil to increase the contrast between rings. And then we had at least 2 independent observers count rings under a dissecting scope. When they didn't agree, we actually had a 3rd observer mediate. So after we did all of that, spent a whole summer collecting data, spent an entire year in the lab counting rings, characterized the stand age structure for these 14 stands. Did we find a stand age structure that was analogous to any of the dominant tree species that we thought may be candidates? And the short answer is no. To be fair, once we saw the sagebrush demography data, we didn't really pursue this line of thought much further, and this is why. Sagebrush was a lot younger than we had anticipated, and by a lot, by decades. So this is all pooled data, so 636 live slabs. Our age range was 0 to 85 years. The mean age of all of those 636 samples was about 15 years. So we took a step back and we thought, okay, did we for some reason oversample seedlings? Had they not self-thinned yet that year? We sampled in about July, so it could be possible that we were still to have some juvenile mortality that we hadn't picked up yet. So we took out everything that was under 3 years old. And when we do that, our average age of big sagebrush only went up to about 16 years. Less than 7% of our sample came from shrubs that were younger than 3 years old. Only 2% of our sample were from individuals older than 40 years old. So going back, that central area of the site, a bunch of it has since burned, but when we did this sampling, these were areas, they've been removed from grazing since 1950 or prior. We know that they haven't experienced fire in at least 100 years. We can see 100-year-old fire scars on the site, and those were in a matrix. So we know that those stands were at least 100 years old, and some of them quite possibly 200, 300, 400 years old. And the oldest individuals over 40 years old only made up 2% of our sample size. We did have at least one individual in each annual age class from 1970 through when the study was conducted in 2006, which was cool. It tells us we have at least some recruitment every year. And we found that there was a course relationship between annual age class and total annual precip. That's what our pooled age class distribution looks like. And just to bring your attention to a couple of things. We do have individuals populating every class back to 1970, but with our mean age only being 15, maybe 16 years old, that empty age class in 1969, it's impossible to tell whether that was a lack of recruitment or simply that part of the population has just died off already. Another thing to look at here, this dip here is 2002, 2003, and 2004. Those are some of our driest years on record. If we look at the precip, there again, it's 2002, 2003, 2004. So less than half average annual precip for those years. So that's one of those cases where recruitment does coarsely reflect precipitation. And here's a case where it doesn't. 2006, when we sampled, compared to the decade prior, we didn't have a lot of recruitment, but it was one of our wettest years. So we went through a lot of iterations of going back through precip data. We looked at timing, we looked at temperature, we looked at snow melt, all kinds of things. And we found that, about half of what explained the amount of recruitment was precip, and the other half was probably local environmental variables, things like aspect and slope and even surrounding plant communities, things like that. So when we break it down to a stand-by-stand level, each of those 14 plots, the mean age of those stands ranged from about 7 years to almost 23. And individuals at the age of death the mean age of individuals at age of death from about 26 years to almost 50. And that's what our youngest stand looked like. Again, this is one, the mean age was about 7 years. And certainly there are youngsters in there, but there are also a lot of mature individuals. There's some summary statistics for that stand. Again, there's going to be a lot of differences here that you see in terms of cover, in terms of density. But one thing that we noticed, mean cover of dead shrubs, so standing dead skeletons, we measured that in line interception as well. You don't always capture this in the pictures, probably because of the oblique angle that you're looking at it, but we always had a fairly substantial amount of standing dead shrub cover, even in those live stands. This one you don't see it as much because there are so many young, vigorous individuals, but we still had over 10% standing dead. That's what the stand distribution for this particular stand looks like. These on top are shown in 5-year age classes and down here in annual age classes. So this population certainly seems to be young and growing. Most of its individuals are in young age classes. Very few individuals out on the tail there. This is certainly a stand that we would consider to be young and healthy and vigorous. There's our oldest stand, and it looks like an old stand. But remember, the age of this stand is still only 23 years. Sagebrush density is down quite a bit in that. That's some of what you're seeing. But this one actually has less standing dead shrub cover than the one you saw previously. And there's the stand age distribution for that one. And again, if you consider the pooled population mean age is around 15 to 16 years, this stand certainly seems to be declining. Our biggest age classes are older than our pooled mean. And we have almost no individuals in those first 3 years, actually first 4 years in the annual age classes. So no recruitment here in the last 4 years. It was really stunning to us, though, that the old end of our spectrum would be an average of not quite 23 years. And we found that stand age structure was very stand specific. in terms of the number of annual age classes that were represented. In the terms of histogram shape, some histograms were really flat. We had no more than 3 individuals in any single annual age class. Some histograms had a nice bell curve, some were multimodal, just across these 14 plots. And if you were to go into each of those histograms for each of those 14 plots and pick out the year with the biggest age class, it was different for nearly every single one of them. And population trends were different. As you could see, some were increasing, some were declining. And those differences weren't always easily explained by spatial context. It turns out that those two that ended up really close together ended up being our second youngest and our second oldest stands. There's our second oldest stand, which again is surprising. There are individuals in there that don't appear to be terribly old. There's our second youngest stand. This is the one that surprised us. It turns out that we have lots of individuals in the 3- and 4-year-old age classes here, but not something that you would necessarily take a glance at the stand and see. Which brings us to the next objective, if you will, of the study. Sorry, I can't seem to put together a presentation without one nasty data slide, and this is it. So up here at the top is the important row, and we did a big multivariate correlation analysis. And what we found is that big sagebrush cover, green rabbit brush cover, the amount of dead shrub cover, that condition rank variable I told you about, or density, none of those things tell you anything about the mean age of the stand. You know, that makes it tough to do diagnostics, doesn't it? There's a few interesting results. Mean age of living individuals was correlated with the age at which they died. At the office we joke this is the live fast, die young theory. Some stands apparently go that way. There's a few positive correlations in the middle here. Those are mostly dealing with green rabbit brush, and they're kind of driven by a few anomalous plots where we had really high rabbit brush cover. And then some positive correlations between density and cover and condition rank, all things that you would expect. So, what did we learn about stand replacement and those large-scale population declines we've been seeing? First of all, we found that turnover rates are very site-specific, but they're much, much higher than we expected. Given that we were working in an area where we know there was no fire for at least 100 years, more for a lot of areas, those sagebrush populations have turned over many, many times since the last disturbance. And we know that population status and stand condition probably fluctuate over time. There was a really cool paper, I think it was a 1948 paper, that talked about sort of this cyclic pattern of big sagebrush stands. They would go from being, you know, very healthy looking to sort of looking decadent, and they would cycle that way over periods of decades or so. So stand condition and mean age may actually be related, but may be decoupled. We know that those ongoing chronic losses that we were seeing in big sagebrush probably weren't a legacy effect from the die-off of the 70s. All of these shrubs are younger than that, or most of them, anyhow. And we know that precipitation patterns do affect recruitment, probably mortality as well, but that was beyond the scope of this study. And that may explain some of our declines. In the decade before this study was done, we only had 3 years of precip that were at or above average. So if big sagebrush is requiring decent precip for germination, it could be that these declines are just a lack of recruitment having to do with low precip. Unfortunately, it's pretty tough to put together conservation management strategies for stochastic events like precipitation. But we do know that disturbance isn't necessary, at least here, to facilitate stand replacement. And in fact, we know that just because a stand looks to be in poor condition doesn't mean that there aren't a lot of youngsters under there. And we know that we can't easily infer stand age from any other variable that we can measure directly or otherwise. And for us, decadence is probably not a terribly meaningful concept because we had lots of standing dead in all of our stands. The young ones, the ones that were increasing, the ones that were declining, all of them had lots of standing dead individuals. So the cooler piece for me is, since I couldn't provide our conservation folks with a whole lot of help, was how does this relate to, what does this mean for sagebrush ecology? And I spend a lot of time talking with our wildlife guys, and they really like thinking about it in terms of it being, of things being either R-adapted or K-adapted species. So you can think of species that are typically slow growing, low reproductive rates, very slow and steady, or things that annuals would be an example of an R-adapted species. They tend to put out a lot of seed and really try to exploit resources that are available in the here and now. And now that I'm almost near the end of my talk, I'll give the disclaimer that I'm actually not a population biologist. I spend a lot more time thinking about physiological ecology. So when I saw these recruitment patterns, they reminded me of the resource acquisition strategy for big sagebrush. It turns out that big sagebrush isn't always a terribly efficient species. When it's trying to assimilate carbon in spring, water use efficiency goes way down, and photosynthetic rates can be astronomical. So it kind of goes after resources while it can. I don't think I touched anything. And it may be that in terms of recruitment, it's doing the same thing. It's maybe not as slow and steady as what we may have originally thought, and it really goes after resources when it can. One thing that we started to see in the data, and unfortunately we didn't collect enough of the right data to address this question, is mortality doesn't appear to be density dependent, which is what you would typically think of in a K-adapted species, that sort of slow-growing, long-lived species that we've always thought sagebrush was. And there's lots of other studies that show that along that R-adapted, K-adapted continuum, sagebrush may actually fall less to the K end than what we had thought. It tends to have early sexual maturity, it produces an abundance of small seeds, and it thrives in environments where resources are discontinuously available. So I'm not suggesting that sagebrush is a colonizer by any means. Any of you who have tried to reestablish it know certainly that it's not a colonizer, but I don't know that it's helpful to think of it as a long-lived, slow-growing, very static species either, because I think, at least what we're seeing in some of these population studies, that the populations are much, much more dynamic and things are happening much more quickly than what we had expected. Some acknowledgements. The funding was from Department of Energy. Wildlife Conservation Society has been a partner in the conservation management efforts. Kurt Jensen led the field crew that sampled this data. Lance Cosberg, some of you may know him, he's local. He was patient enough to sand all 915 slabs 3 times. Kurt Edwards and Jeremy Shive provided GIS supports, and Doug Halford reviewed the manuscript for this, which is not available yet. And a lot of this work started 10, 15 years ago with talks that both Roger Blue and I had with Jay Anderson, so in some ways this was a bit of his brainchild. And this information is available in a technical report. I was hoping it would be out by now, but sequestration has caused our grinding to go even slower, so it's the report's complete, but it's working its way through the cogs of DOE right now, and I'm hoping it will be available on this website sooner than later. If you're interested and want me to let you know when it's available, catch me, give me a card, and I'll be happy to email you a live link when it comes up. So with that, are there any questions?
We have time for one question for Amy.
Did you look at
Just a second.
The question was, did we look at
recruitment and cheatgrass? Is cheatgrass an issue for you?
The question was, did we look at recruitment and cheatgrass? Some of those plots had more cheatgrass than others. It certainly wasn't a dominant, and it wasn't even the most abundant herbaceous species. So most of those plant communities were what we would still consider decent condition native plant communities, although cheatgrass is present in most of them. Any others? Nope, only had time for one. Okay, thanks.